The story you're about to hear is a compilation of documented true facts about historical characters, events, or locations. Please sit back and listen as I narrate this story to you. The role of the U.S. Food and Drug Administration in the regulation of novel medicines arose from tragedy. In the fall of 1937, 71 adults and 34 children died after taking a drug called Elixir Sulfanilamide to treat a variety of ailments ranging from gonorrhea to sore throat. At that time, the FDA, which had been founded in 1906 as the Bureau of Chemistry, was primarily responsible for policing claims about food and drug ingredients. To market new drugs, no formal government approval was required. As a result of 27 years of public awareness in the United States of numerous problems with food and medicines, such as syrups for infants containing opium derivatives, nostrums and panaceas were widely advertised to cure a variety of ills. Dr. Harvey Washington Wiley, a government chemist, played a key role in the passage of the 1906 Act in 1902, launching a widely followed study of the effects on human volunteers of a controlled diet supplemented with various preservatives in widespread use at that time. A group of 8 young men, dubbed the Poison Squad, were allowed to eat dinner with him and then take capsules of various preservatives in use at that time, beginning with boric acid, then copper sulfate, salicylic acid or aspirin, and formaldehyde. Needless to say, this public demonstration of the dangers of food additives prompted Dr. Wiley and Alice Lackey's work to inspire 1 million American women to write to the White House in support of the 1906 Act. The Bureau of Chemistry, led by Dr. Wiley, was given jurisdiction over the enforcement of the 1906 Act, and the FDA was established years later. The 1906 Pure Food and Drug Act was initially concerned with product labeling, but was later expanded to require certain drugs, such as alcohol, cocaine, morphine, and cannabis, to accurately indicate dosage. The 1906 Pure Food and Drug Act needed revision by the 1930s, and it languished in congressional committee as disagreements over its replacement arose. In the 1930s, sulfanilamide, a member of the sulfonylamides or sulfa drug class, was discovered to be effective against Streptococcus bacteria. The origins of Elixir Sulfanilamide can be traced back to German dye industry. Paul Ehrlich, the visionary doctor who developed the first reliable syphilis treatment, began his career by researching how to stain cells so they show up better under a microscope. Ehrlich discovered through careful experimentation that some dye molecules, the same ones used to color garments, only stain bacteria. This prompted Ehrlich to propose a radical theory. What if it were possible to dye molecules so that they would only attack bacterial cells while ignoring human cells? A potent antibiotic could be the end result. Ehrlich devoted much of his career to attempting to answer this question. Despite his success with the arsenic-containing atoxyl, doctors were wary of its proclivity to cause vision problems in patients. Nonetheless, Bayer Laboratories chemists remained confident that dyes would reveal the world's first true antibiotic. Bayer and its parent company, IG Farben, were dye specialists. Joseph Clarier and Fritz Mitch, its chemists, had special expertise in meticulously tuning and changing their properties. The Bayer team created thousands of different dyes and tested them on Gerhard Domagk, a doctor who tried, often unsuccessfully, to cure bacterially infected mice with them. Domagk tested a red dye that caused complete recovery in mice in 1932. Prontosil was the drug in question. He went on to win the Nobel Prize in Medicine in 1939 for discovering the antibiotic that saved the life of Franklin Delano Roosevelt Jr. Further research by a French team discovered that Prontosil splits in half after patients take it, and that only one of the 2 pieces is effective in fighting bacteria. This small fragment, known as sulfanilamide, was just as effective at curing diseases as Prontosil. Even better, the patent protecting it had expired years before, allowing anyone to create medicines that used it. Following this discovery, Bayer attempted to sell a version of sulfanilamide, but discovered that the challenges of competing with generic manufacturing companies made it unprofitable. One of sulfonylamide's greatest strengths, derived from its dye heritage, was also one of its greatest weaknesses as an antibiotic. It refused to dissolve in water. A good dye should not wash out of clothes or carpets, but it is much more difficult to get people to eat a dry powder than a spoonful of medicinal syrup. Sulfanilamide was simply too difficult to consume for it to be widely useful in medicine, particularly for children. The founder of S.E. Massengill Company, Samuel Evans Massengill, set out to solve this problem. Harold Watkins, his head chemist, was challenged to create a sweet liquid form of sulfanilamide. Watkins discovered that a solvent, diethylene glycol, could dissolve buckets of the stuff after much experimentation. It was a 10% sulfanilamide, 72% diethylene glycol, and 16% water solution. The solution's appearance, taste, and fragrance were approved by the company's internal control lab. It was flavored with raspberry extract, saccharin, and caramel, among other ingredients. And by September 1937, Massengill had distributed 240 gallons of the liquid known as Elixir Sulfanilamide across the country. At that time, there was no regard for testing or safety or toxicity, and over 600 bottles were distributed to pharmacies and physicians throughout the Southeast, mainly in Oklahoma and Mississippi, although another 700 bottles were distributed to physician offices and Massengill sales representatives as samples. Diethylene glycol, or DEG, has a lethal dose of only 2 ounces. For those who had consumed the mixture, nausea, vomiting, and abdominal pain were often followed by kidney failure, weakness, and coma. The first telegram to the manufacturer was sent on Saturday, October 9, 1937, from the American Medical Association office in Tulsa, Oklahoma, reporting the first 6 deaths with similar symptoms of low respiration, kidney failure, and lower than normal temperatures. By the following Monday, the death toll had risen to 11, with 2 more being treated for gonorrhea with the elixir of sulfanilamide. The Tulsa AMA discovered that the solvent used to make the elixir was 60 gallons of DEG, with no known antidote or treatment for DEG poisoning at that time. FDA Commissioner Walter Campbell immediately directed an investigation by the vast majority of the agency's 239 inspectors and chemists. Francis Oldham Kelsey, a tenacious FDA scientist, identified diethylene glycol as the poisonous component of the mixture, which Watkins, Massengill's chemists, had not realized when creating the concoction. He became increasingly overwhelmed by the rising death toll from his creation, and he committed suicide less than 2 years later. On Friday, October 15, 1937, a week after the initial report, the shipments ceased, and the company issued 1,100 recall telegrams. The death toll had risen to 22 by Friday, October 22, 1937, and the FDA dispatched their entire staff of 239 field inspectors to search for every ounce of an unaccounted-for elixir. In 1937, there was still little electrification in the southern United States, and many people did not own radios. Newspapers were the only means of mass communication, and the story was buried inside. On Monday, October 25, the death toll reached 41, and it was announced that nearly all the bottles shipped had been collected. 95% of the 240 gallons produced were either recovered or otherwise accounted for. However, there were a total of 105 confirmed or probable deaths because of this tragedy, with 37 of these victims being children. The youngest victim was only 6 months old. Massengill was summoned to court to answer for the disaster his company had caused, but he was unrepentant, and he was correct under the laws of the time. Massengill hadn't done anything wrong. Analysis of the Tulsa patient's concoction revealed that the ingredients were exactly what the company had said they were. There was no requirement to demonstrate the safety of new medicines. In fact, the FDA's only requirement for new medicines was that they be accurately labeled. Massengill was prosecuted to the fullest extent allowed by law. He had used the term elixir incorrectly on his medicine. An elixir must use ethanol as a solvent, not diethylene glycol, and he was fined $26,000, which is now worth more than $500,000 in today's dollars. Even though it was legal, The Elixir Sulfanilamide incident sparked public interest in drug regulations to prevent such tragedies. On March 5, 1938, Senator Royal S. Copeland of New York responded to the outcry by introducing the Federal Food, Drug, and Cosmetic Act of 1938, or FFDCA, to the Senate. After 2 frantic trips through both houses, President Roosevelt signed the final FFDCA on a hot June Saturday, enshrining in law the FDA's right and responsibility to demand the safety of new foods and drugs. Elixir sulfanilamide took the lives of 107 innocent people, but it saved countless others from a similar fate. The FFDCA protected the public from dangerous medications, but its limitations were quickly revealed. For one part, it gave the FDA only 60 days to prevent dangerous drugs from being sold. For another, it did not permit the FDA to ensure the quality of the medications or even require that they work as advertised. However, it wasn't until October 1962 that Congress passed the Kefauver-Harris drug amendments, which required companies to provide evidence of efficacy as well as safety for drug approval. This act would be amended in 1976 to include medical device regulation by replacing the Pure Food and Drug Act of 1906 and giving the FDA significantly more powers and authority. This is where we can find Section 501(k) for the 90-day notification of intent to market a medical device, the different classes that devices are approved under, and information on the pre-market approval or PMA process. The FDA was founded on both outrageous claims and a medical tragedy. This history is critical to remember when considering the FDA's mandate for the safety and efficacy of all medical devices, including in vitro diagnostics. Hey everyone! I just wanted to express how grateful I am that you took time out of your day to listen to my narration. This is Nikki of Twysted Mind, and I'd like to wish you a wonderful rest of your day. Salamat!